Thank you very much for the kind introduction and also for the opportunity to talk about some of our work. As Anton mentioned, this is something new which we hadn't really thought we would ever work on. But I guess one of the privileges working at the Max Planck Institute that you can sort of follow up things for which you don't have, grant funding. You can follow leads, and it's actually a principle of research at Max Planck Institutes that you shouldn't do things which are obvious and doable and fundable, but you should take risks on new things which may or may not work. And of course, that's easier said than done. Obviously, I don't do this work myself, so you also have to find postdocs or students in this particular case and colleagues who are actually willing to share the risk of new things. Normally, the first slide is kind of bypassed very quickly by speakers, but that first slide is sort of, together with the second slide I will soon show you, phrasing the concept about what I'm going to talk about and also being a take-home message. So what you see here are these funny trajectories, lines, right? You see, yeah, lines that yellow lines that go down, go into reddish orange lines that make a turn towards the right, and then go into purple lines that go up. These purple lines change direction, and color coding direction become orange, reddish, and so forth, right? A very complicated thing here, right? Many lines, not random, crisscross, but in certain directions, but complex directions. We'll tell you what these actually mean. Now, what you are certainly familiar with is images like that. This is from your town here, the port area. So I took this because my hotel is nearby. Here you see again lines, some are roads which make turns, or as you go on the highway go in and out. There is of course also the port here with the various channels that direct ships to go into these particular areas where they are loaded and unloaded. And then of course there's also, I guess this is a train station here, with trains coming in a certain direction. Now again, this is not a crisscross, right? There is order in this, right? Obviously, let's say if something comes from a boat and is brought, let's say, furniture from somewhere else, is unloaded here in the docks, right? And then put on, let's say, a truck, that then goes onto the highway and transports this to, to another place in Bulgaria, or it's unloaded onto a train and the train brings it somewhere. So there's a complex traffic pattern here, right? And such things you find in all sorts of human activity. So if you, for example, look at the internet traffic, it also is sort of like that, right? You think it's random, but of course, clever and now famous companies like Google can track that pattern of your searches on the internet and can decide whether you should go on a truck or your message or a query should go on a truck or on a train or whatever, right? And there are hubs and nodes that organize complexity. And what I wanted to tell you, and this is the title here in the brain, we have actually such roads in addition to the neurons and nerves, which are now studied for more than a century. We have other roads in our brain, and those are roads that are deployed in the ventricles. We are not working on human, but you are here. This is a medical school, so remember, just remember that the ventricular system in the human brain consists of the lateral ventricles, which are C-shaped here, quite large. I mean, a human brain is several 10, 15 centimeters, 20 centimeters. So these are centimeters here, right? And the lateral ventricle, of course, drains into or connects to the third ventricle through the foramen of Monroe into the third ventricle, which is in human as well as in rodents consisting of two parts here, a dorsal part and a ventral part. Ventral part goes deep into the hypothalamus, as you know, connecting down to the pituitary. And then, of course, the third ventricle drains through the aqueduct into the fourth ventricle, and then this is filled, as you know, with cerebrospinal fluid, and the cerebrospinal fluid is thought to come from, from the from the lateral ventricle, third ventricle into the fourth ventricle and drains in the subarachnoid space. So this is the way it's supposed to be. What is interesting is, and one can already see it on that slide, is that this is, just a mold here, but when you look at the inside of the walls of the ventricles, they are coated with ependyma, a special epithelium, and the great thing in these ependymas is that they are actually ciliated. The cells are ciliated. This is actually, I think it's from lung, but it would look relatively similar in the ventricles of the brain. Cilia here are these long things, whip-like things. Cilia means eyelash in Greek, and see these 2 microns here, right? They are only about 200 nanometers in diameter, but in the ventricles about 15 microns long, so very long cilia. And they, not uniformly, but they coat the inside of the ventricles. In addition to, of course, having cerebrospinal fluid in there. And it is a discovery that the ependyma is ciliated, which goes quite some time back. It's a German journal called the Archive for Anatomy and Physiology and Scientific Medicine. This is the publisher here, and this is a volume that appeared in 1836. And I have this front page because there's an interesting paper in here, 2-page paper by Purkinje. Purkinje, of course, is known to all of you more from the neuroscientist side, but he writes, and he wrote here in German, that he, say first in German, gelang es mir endlich, den 28. Mai die Flimmerbewegungen an einem sehr wohl bewollten, ziemlich reifen Schaffötus, und zwar den anderen Tag nach dem Schlachten, etwa nach 30 Stunden am Rande der Fimbria und so forth, die schönste Aktivität zu entdecken. So what he says here, he got he saw on the 28th of May 36 in a rather advanced fetal sheep. Bewollt means had already fur, right? And he saw in that, opened up this brain, and he saw at the margin of the fimbria, that's where the hippocampus is close to the lateral ventricles, 30 hours after the fetus was collected, wonderful flickering activity. I don't know how he saw that. These cilia are small, but he saw that in the ventricles there was a flickering. And he realized, and this is here on the next page, that this flickering was caused by little thingies here which pigeon-förmig, whip-like, moved and had at the end, at the cellular end, little granules. These are the basal bodies, but so it is totally surprising, how careful observation allows you already 200 years ago to make fundamental discoveries, right? So he discovered the cilia, beating after 30 hours after the brain was collected in a sheep. So another, that was 130 years later, again is sort of, Anton was saying CSF and ventricle is sort of a little bit obscure in science. It's not like, Nobel Prize generating kind of research at this point in time. So this Worthington Cathcart, 1966, so where they published, this is now not a human brain, as you realize, it's a rat brain. So this is the third ventricle here outlined. It's a sagittal section through the middle. So the ventral part of the third ventricle. And what they did, these were sort of thick slices. They put red blood cells into the medium. And they saw that these red blood cells somehow moved from the entry duct of the third ventricle, made some sort of funny movements, and then exited at the other end. So they used red blood cells to see that, right? So that was, to my knowledge, the very first time anybody has talked about visualizing the fluid movements in the preparation of the third ventricle. So of course they had assumed that these would be the Purkinje type cilia that would do that. But that is pretty much it. There was a Japanese paper, I have no pictures here, scanning electron microscopy, and they saw, again in the rodent brain, that these cilial bundles had certain spiral orientations. Of course, you know, that's a fixed preparation, you can conclude anything from that. But anyway, at this time, this work and the Tamadori, the Japanese work, suggested that maybe these flows of CSF are not so uniform. But the general idea still today is CSF comes in, goes through the ventricular system to flush out things the brain doesn't like. You know, you can find, for example, amyloids in CSF and all sorts of cellular debris. So it's like a flushing system, like a toilet. But let me one sec. Yeah, maybe this is useful to see. I have to activate the movie here. This is now some pictures we did, just to give you an impression. So this will be interference contrast images, slow motion of cilia in the third ventricle of the mouse. So the resolution of this project is not fantastic, but you can see that there is movement here, this sort of like flickering, I suppose. And what this actually is, that, as I said, it's like a whip, right? So if you look at the movement of the cilia from the side, it sort of goes back, but it doesn't go back straight, but it sort of goes back this way, like a whip, and then goes forward this way, right? But anyway, this is a lot of movement here, and this is a picture, a sketch of a cilium, right? It's an organelle, it's attached to the cell, covered with plasma membrane, and if you do a cross-section, you can see it has this typical 9 pairs of microtubules at the periphery and the central pair of microtubules in the middle. And there are lots of proteins that migrate or move on the microtubules, including molecular motors, the dyneins, the inner and outer dyneins. So I don't want to explain how this whip-like movement actually occurs, but you can imagine, simple-minded, that the molecular motors move the various duplets of microtubules and thereby create this whip-like movement. And the frequency of this is temperature dependent, but around room temperature it's about 30 times per second, 30 hertz, 40 hertz, depends also a bit on the tissue. So this is of course slowed down, one wouldn't see it in real time. So, good. Coming back to the ventricular system. So this is a cross-section, I think it's the last introductory slide, through a mouse coronal section, through a mouse dorsal, right, so hippocampus here, the third ventricle. And the CSF, cerebral spinal fluid, has 2 origins, but the major origin is actually the choroid plexus. So the choroid plexus is this secretory epithelium with tight junctions, which forms these sort of very lobulated structures. So this is a high power here, and this is the dorsal part of the third ventricle. And you see that the choroid plexus is in here, extending essentially along the entire third ventricle. There is, of course, also a choroid plexus in the lateral ventricles, even larger than this one, and in the fourth ventricle. So this is the major site of CSF production. Some more CSF comes direct from the brain parenchyma. And CSF is not a boring salt solution, by no means. I mean, mass spectroscopy done in the last 5 to 10 years has shown that there are hundreds, maybe thousands of compounds in there, not only small molecules, but also peptide hormones, proteins, of course also things like waste. I mentioned amyloid in Alzheimer and Parkinson patients. So this, we have looked, for example, at a transcriptome pattern of this coil plexus here in the third ventricle, and that expresses hundreds of thousands of genes, and hundreds of them encode secreted proteins. So the CSF is something highly interesting. So what I like to, after this introduction, what I like to discuss with you today are questions and issues. Issues is sort of a way, I know it's interesting, but I really don't know what it means. So we know from this classical work and many other pieces of work that have published after Purkinje, and so Celia promotes CSF movement. But is this to simply flush through CSF and thereby remove debris from the brain? That is, the common dogma. I don't think that's the case. CSF is rich in signaling compounds. How are these delivered to their target sites in the brain? So this is the Warner map issue, right? So how do you make sure that the wine goes onto the right boat, right? It's a non-trivial issue, right? And there's not only wine, there's many things that are produced here, and so it's like the very rich CSF. Now, of course, we know from the neuroanatomy that behind the wall of the third ventricle, behind the ependyma, this epithelium coated with cilia, resides a complex system of hypothalamic nuclei that coordinate the control for a variety of critical body functions. So it could be, and it's thought, hypothesized, that substances in the CSF somehow affect the function of hypothalamic nuclei. The ependymal lining in the third ventricle of the brain has locally enriched population of Tannis sites that have elongated processes extending deep into the hypothalamus. So there are cells, in addition to the ependymal sites, the Tannis sites they are called, in special locations, not throughout the third ventricle, that have their cell bodies, is a form of glial cell, exactly at the ependyma. So they have direct contact with the CSF, they are not ciliated, and then they make, if you will, like axons, very long extensions that go sometimes millimeter deep into the brain parenchyma, including the various hypothalamic nuclei. And of course, we also know that the ependymal cells are actually very loosely connected. There are no tight junctions. So the brain is able to secrete through parenchymal flow substances it creates. This could be peptides or neurotransmitter. The point is, what I just said before, there's a complex logistic task. There's a fluid that has many compounds. There are potential target sites. There are different target sites. How do we organize the logistics of all of this? And if it's just flushing through the ventricles, even with an occasional whirl, that's not going to do the business, right? The port of Warner is also complicated as other ports. But this is just to, I showed this diagram of the third ventricle because I am using sort of terminology throughout my talk, which I introduced here. So I mentioned there's a dorsal part and a ventral part. So there's coil plexus only in the dorsal part here along the roof. So cerebral spinal fluid comes in, first of all, from the lateral ventricles, which are not in this plane through the foramina of Monroe. So there would be entrance here, but because it's a mid-sagittal section, you haven't drawn this. But so then here we have CSF coming here, eventually going through the aqueduct. But then there is also a relatively narrow bore tube that goes from the dorsal third ventricle down to this ventral pouch. I call this sort of a pouch, or it's like a lady's bag with a handle. See, the other handle is here, which is the outflow, right? So there is flushing or whatever I show you, right, of CSF here. Just as another orientation, I mean, here this is from a mouse, and these are millimeters, right? You probably know there is a sort of a ruler in the mouse brain called bregma, right? And so this is 1 millimeter. So we are talking about dimensions, these are also millimeters here in the vertical, of several millimeters. In human it's a bit larger, but it's not orders of magnitude larger, but it's larger. In a rat it's somewhat larger. There's very similar structure. So here again are these a few of the hypothalamic nuclei that are either directly contacting or very close to the ventricle. So like the suprachiasmatic nucleus, the paraventricular nucleus, and so forth. Down here is the median eminence. As you probably know, this is a critical organ for endocrine function of all sorts. So what we wanted to see is, look at the flow in these different regions, right? The first thing we are seeing is we're looking into this dorsal third ventricle from above. So the way we do this is we have a relatively simple setup. This is a microscope, can be equipped with fluorescent interference, differential interference contrast, has a fairly sensitive fast camera on top of here, and computer, right? So it's quite simple, right? And so what we can do is we can look at this third ventricle from top. We have to take the mouse brain out, and then we have to remove the cortex and so forth, right, carefully. And then we can just look down into the third ventricle, make a small incision, and look what happens in there. So we have two movies. One is in a plane, roughly at the roof, and one deeper in, almost at the floor. So as you see, we'll see it's very simple what's happening here. So here, the dorsal level, right? So you see, basically, this is the axis of the third ventricle, the dorsal part. So this is this axis here. This will be rostral caudal, and you see the beads basically flow back, right? These are small fluorescent beads. That's why we use a fluorescent microscope, 1 micron in diameter. However, very surprisingly, if you go deeper in your focal plane in a microscope down at the floor, the beads go just the other way around. So they don't go to the aqueduct, that they go this way, right? Or to this point of the entrance of the anterior entrance duct. Now in the next movie, let me just go to the next one. We are actually looking here. So now we are at the roof of this pouch, of this ventral part of the third ventricle. And although it's not a fantastic resolution of this beam, but you can clearly see that beads come here from the back. So this will be from caudal, and others come from here. And then what happens? They sort of go sideways all the way down deep into the back, right? So this is again, of course, not telling us that basically the whole thing is flushed through. There are currents, even in a very limited plane, that go this way and this way, come together and go down. Right. This, right? We call this, you will see a bit more later from another perspective, separatrix. This is a fluid dynamics term. When fluids come to each other and then confront each other and then go down into the volume, they basically, what that means is it separates this volume in two halves. There is no way a substance from here can get to the other side, and nothing can get from here to this side. So this is called a separatrix, a separation current, right? So this is not what one had expected to see. Now, to document these currents more thoroughly, of course, they're three-dimensional. The Holmans are not that straightforward. We cannot see down deep into the pouch. So what we do is we take this pouch and cut it in the middle and flip it open. So it's like opening a book. So you have the two walls of the ventricle, and you can, with the microscope, directly look from top, right? So this is what is happening here. So let's just look at A here first. So this will be your tissue piece, one of the two walls. So these are quite sizable, several millimeters, but remember, Pocringi already knew that this works very well even after 30 hours after you have killed got the sheep fetus. Cilia here, and this is filled with a buffer, so the water buffer is very critical, but there's nothing magic really. And then these dots here are beads. So these are the beads you saw before, 1 micron beads. You can also have smaller beads or larger beads. They are so small that they float here. And then of course the cilia here beat with frequencies of 30, 40 hertz. Microscope at relatively low power, 5x or 10x, looks from above, and you can see these beads moving. And you know, it's like a plane in the sky. If you have maybe a very busy day, many planes in the sky, and if the weather is such that they actually have one of these condenser strips behind from their exhaust, you can actually follow them, right? So there are mathematical procedures you can use to track the beads to make these sorts of trajectories the plane has, right, in the sky. And this is what you see here. So this is, you know, the picture I showed you at the beginning. So these are beat tracks visualized by a beat tracking program. And here, you know, to orient you, this is the anterior commissure, this is the entrance duct into the ventral pouch of the third ventricle, and not shown here, but this is the exit. And we have simply color-coded the direction of beat movement. So yellow means towards 7 o'clock, if this is a clock face, so 7 o'clock, 1 o'clock. So you can see it comes in here, behind the anterior commissure, goes down into the most front anterior part of the third ventricle, then turns around, comes up. So you can see, for example, here 3 currents, 1, 2, and 3. Not all beats that come in go this way, but some go directly straight back. And this is this area where current 3 and current 4 confront each other. This is the separatrix, right? So you see the separatrix exactly here as you saw it in the Holman, right? These guys here move here, and these move here, and there's no way they can cross, so everything is pushed down, and you see push down means, you know, towards 6 o'clock, 7 o'clock. And this current here coming in, 3 goes over in 5 ventrally, and then goes down here. Here is another sort of an interesting movement. This is towards the posterior end of the pouch, goes down, right? And next to it, it goes just the other way, right? Some of it is caused by turning, so these beads move up, down, and up to the exit. And these guys here come from underneath here. This is actually a dead zone. It's black, right? This is the area where we have lots of tanycytes in the third ventricle. These tanycytes, as I mentioned, have no cilia, but they have these long processes that go to the nuclei. Behind here is the arcuate nucleus, as you may recall, and the median eminence. So, you know, this is of course not something happens in the vacuum, but there are all these nuclei behind. So very complicated pattern of movement, and of course in a net way nothing can get lost, right? Fluid is not lost, right? So everything is connected, like when you have a congestion on a highway, eventually will you know, be solved, but sometimes you have to take a detour. Now these are 1 micron beads. You say, well, you know, there are no 1 micron beads in our CSF. This is just about proteins and peptides and whatever you have. But of course, these beads represent the flow of CSF. But you being critical, of course, you can ask me, show how it is when you have a protein, right, rather than a bead. So I don't have a protein, but I have a polymer, which is called FIT dextran. And what we do here, let me just go back. What I'm going to do, I have a very fine needle and inject fluorescein dextran. This is 70 kilodalton fits FITC labeled polymer. And I inject it at 3 positions, so the needle tip is at position A, B, and C. And remember, the beads in A go down this way towards, let's say, 7 o'clock. In B, they go pretty much straight down. And in C, they go towards 3 o'clock or 4 o'clock, right? Let's now inject at these 3 positions and look what happens to Fitz dextran. So this is the first position, goes down and caudal, right? Like the beads. Now the needle is soon be in position. Here this guy goes down And look how it gets spread. So it's very different, right? But it's exactly what the beats are. And then in the third position, here. Right? And the rate of this movement, this is just a repetition. The rate of this movement is in the range of, let's say, a couple of 100 microns per second. And the whole pouch is about 3 millimeters, right? So nothing goes from one to the other end, but the dimensions we are talking about transport is a millimeter range. And the rate is maybe 200 microns per second. So these are real speeds that are realistic, right? Are useful transports. It's not like a snail or so. It's not as fast as neurotransmission, but of course what the nuclei in the third ventricle, around the third ventricle do is not neurotransmission, but they regulate things that take time, like circadian clock. And you know, what is also very nice to see here is that depending where you inject, the material stays together. It's like a slug moving, right? It's not that it spreads right away. But there are places like here where this spreading happens reproducibly, or even down here as well. So it looks as that there are sort of ways of distributing things, less or more, or keep them together. I should say, and I sort of already said that before, that the advantage of these preparations is that they are actually very persistent and constant. So if you have the right buffer, when you, 10 minutes after dissecting the brain out of the mouse, this preparation is in the dish, and that's the way the pattern of movement looks. If you look 5 hours later, it's the same pattern. There are some spots here, these are just beads that got stuck. If you wait another, whatever, 5 hours, it exactly looks the same, right? So this is sort of like what Purkinje said about that sheep, right? It's quite robust. It's not something which goes as soon as you kill the animal. This is experimentally, of course, important, right? Now, the question, of course, is why is this moving this way, right? And the hypothesis would be, yes, this is driven by cilia. So the cilia underneath there, and they are beating in different directions, and they are such directions that create these trajectories. Again, the sort of common wisdom is the cilia all go this way, right, through the entire ventricle and third ventricle, and you want to get just rid of things. Now, you saw that cilia movie before, and it's very difficult to read these movies, and the third ventricle is actually quite large in terms of visualizing cilia, because to visualize cilia, go a few slides back, you now have to take a 30, you know, a 50, 60, whatever X objective. You have to do the oil immersion, and you have to do this now for a piece of tissue which is millimeters in size, right? So you have to collect many individual areas and stitch them together, right? It's sort of what some of you guys do when they make, you know, the scanning microscope. You can only see this at high power. And so you would have, you know, movies that cover hundreds of little images, right? And you can't look at those, right? So you have to have an analytical program that visualize the movement of cilia. And I don't want to go into details, but the cilia bundles, it's not an individual cilium, but it's bundles of cilia that move in a concerted fashion. And then when you take interference contrast images, you can basically, at, you know, let's say for 10 seconds, with a frequency of cilia beating of, say, 30 hertz, you can average the cilia movement by subtracting pixels. Right? We don't need to go through the math of this, but what you clearly see here, so this will be a picture for movies analyzed this way, and you can see these are cilial bundles, right? Not individual cilia. This is sort of capturing the dynamics of cilia beating for 10 seconds, right? And you can see that one can then draw little red lines, as I have done here, right? Which are, you know, marking the direction of the cilial bundle beating. And you see that there are basically two types of directions. Those on the right side here, which would be posterior caudal, are sort of beating all like summarized by this sort of turquoise arrow. And those on this side here are beating towards 5 o'clock like this blue arrow. So like here and here, this is the separatrix, right? So we saw that the beats would come together and confront each other and escape right down into the depth of the third ventricle. And this is exactly the way the cilia would direct them, right? So this movement of beats or fluorescent dextran is really driven and organized by an organized pattern of cilia along the wall of the third ventricle. So this was for the separatrix. Here is the entrance. Remember, there were sort of 3 flow directions. I also showed that with the fluorescent dextran. Here we have again this analysis of cilial beating. Some here in the front go this way, general direction, right, like the blue arrow. Then those here go back, right, like this arrow here, greenish. And then some few go down here. You can very clearly see even here these lines, right? These are these pixel calculations. And this is, of course, exactly how the flow goes, right? The cilia predict exactly the flow of beads and fluorescent dextran. One last case, which I like a lot. This is sort of in the middle of the third ventricle. The cilia organized like giving a circle, right? Like a traffic circle, right? And when you actually look, and this is fascinating, and I hope I can show you that. No, you can't see it. It's just the resolution of the beamer is not enough, but you can come to me later. But basically, you see them organized so that you get a whirl here, right? It's very nicely visible on the screen here. So this is actually real time, right? You can see more down here. So they're very quick, right? Maybe 30 hertz or 25 hertz, depends on temperature. So anyway, so even that whirl is, and you will see actually beat tracks of the whirl shortly. So what we can say then is that this whole logistics of beads and fluorescent dextran and CSF components, that logistic is driven by the cilia, right? And I have said that once you put a piece of lateral wall in culture, in this medium, it doesn't change. So that means it's a program there, right? That is encoded for life, presumably. There's some variation, I'll tell you later. But that drives this program, right? It's like, you know, the highways and the train tracks. Those are not changed, right? The thing in logistics is not changing the tracks. That would be too expensive, right? It's moving between tracks, right? So just coming to that point, so this is our pattern with which we are now familiar. When you cut this, let's say, in 3 pieces, and then pull the pieces apart, and then do the beat tracking on each individual piece, you see already in the individual pieces, it looks very similar to the whole, right? So it's not so that beats are accelerated here and therefore they move in the same direction on the next side. When you pull these apart by a millimeter, the pattern on both sides completely match, because you can take these pieces with your Photoshop and put them together, stitch them artificially together, and you get the same as here. It's a little bit more jagged here because there was time in between and some of the beads got stuck, so that's why you have black areas, but it's very, very similar, right? So that is a very nice confirmation that this happens autonomously. It's not driven by the flux itself. Here is, you know, how do you construct these sorts of things, right? So what was done here is looking at some of the traffic rules. So here we have one piece of a pentamer with cilia on, which is surrounded here with white dashed lines, and these are yellow dashed lines. The second one, the cilia here beat in the same direction. So if you inject fluorescent dye here, fluorescent dextran here, you see this is a minute later. This is a pipette. It moves here, right? And there is a boundary between the two. So here it's at this boundary. And then it wait a bit, and then the cilia beat in this piece upwards here. It goes in and moves up, right? So it goes from one piece to the next, follows exactly the orientation of the cilia. Here, so this is keeps things tight, right? The flow tight. Here is a spreader, so there are two pieces selected and dissected out of the wall of the third ventricle. This piece here, right, and the other one. So you see that the injected dye moves here, then it sort of goes to the other piece. See, this is the boundary of the other piece. And rather than going like here upwards, it starts spreading out, right? So here that's the different kind of, you need to not go straight ahead, but you are at a stop, and then you spread out your goods in, from a big ship into 5 different trucks, right? Not on another ship. And here's another example here, when you have juxtaposition. So you take two pieces whose cilia move in opposite direction, right? Put this piece of tissue, put some dye on here, right? Then you can see it moves, right, like here. But then here at the boundary, I show you the movie, it goes out of focus. This is because it comes together and it escapes on top, right? Let me just show you the movie for that. So juxtaposition. Here this is exactly the same data I showed you on the previous slide. So that one piece, the other piece, so the tissue on this side moves the beads in this direction. This guy moves in the other direction. Here they confront, and they eventually go out of plane. And here is the dye injected. So this will be the needle here. It's exactly the same tissue. Let's see, the dye comes in, moves. I do this with the very straight, very amazing, right? And then here, it's here, right? Goes out of focus, right? Can't go to the other piece because it's a countercurrent, right? So this is sort of, building your lateral wall ventricle of choice, right? Building your own transport system. I showed the circle. You didn't really see it well, but I have been saying that the movement program, the trafficking is constant, right? You can, from mouse to mouse, you can take 2 different mouse strains. There's no difference. However, of course, life is not that simple. I also work on circadian clocks, and so we were looking at whether there's a difference when you collect the tissue, collect the brain, and make the preparations. So what you saw before was all at the beginning of the activity phase of the mouse, which is the beginning of the night, and this at the end of the night. That is where you see this circle, right? So that circle we don't see at the beginning of the night. So you see that this goes towards, 11 o'clock, right? And then this goes towards 7 o'clock, right? Then here this guy goes towards 5 o'clock, it's red. And this one goes, this is purple, right? Towards 1 o'clock. So this is that circle, which didn't show very well. But so this you find only at the end of the activity phase. And of course what this does is when you go back to the other picture here, where there isn't such a circle in that position, this is the circle, right? The circle being here will interrupt the flow for 3 into 5, right? So it's sort of like, okay, here we suddenly change the rules and you can no longer go down here, right? Which is towards the ventral medial nucleus of the hypothalamus, right? So you change the flow game, right? And this, we don't know what it means, but it could certainly mean that depending on the body's clock or physiological activity, you need to redistribute substances. What I have also here, but which I don't show now, there is also some change in aging. Again, the changes are not dramatic. They are subtle, but they redirect flows. So let me come to I think the second last slide or so, to the point here. So this is the problem we started out with, right? So we have to somehow deal how to organize the traffic of substances in the ventricles. And it's an interesting problem because there are many substances, there are many targets where they have to go or where they have to be picked up. So it's a logistics problem, right? And we are beginning to understand the rules, and we are beginning to understand how this might work. We have no idea what happens if you don't have it anymore. Yes, if you don't have cilia, then you have hydrocephalus, that you know, but that's too severe an impact. But I have put in this busy slide here some of the main sort of conclusions, ideas, and hypotheses. This is all new, so we are still very early. So we believe it is important that substances are delivered to the proper location within the ventricular system, not only the third ventricle, but I focus on the third ventricle. To accomplish this, substances are transferred by a cilia-based trafficking system that provides alleyways capable of precise transport. These slugs and these pathways are less than a cell diameter thick, so this is very precise. Rates of transport of CSF constituents, modeled, for example, by this fluorescent dextran, are in the range of a couple of 100 microns per second. They are sufficiently rapid for transporting molecules over substantial distances within the third ventricular cavity, and fast enough to regulate hypothalamic nuclei-controlled functions. So the time scale is perfect. And it is not redundant, of course, with neuronal signaling, which is much faster. Cilia movement creates boundaries that lead to compartments within the third ventricle. So the cilia movement is not only there to transport things, but is also there to, the separatrix is an example, the circle is another example, to make compartments. Right? There are distribution centers, if you will, post offices that can fan out cargo or sort them via circular whorls that could be hubs. So you saw that there were ways where things spread and places where things remained coherent. Cargo could be peptides, proteins secreted by choroid plexus epithelium, released by ependymal cells or tanycytes, or simply leak out of the brain parenchyma through gaps between ependymal sites. So I think what is being transported, the barrels or the casks of wine and the sacks of grain, we don't know. This is, we suspect, this is the current research we are doing. Cargo could be taken up by receptor mediation, so it could be receptors on the ependymal sites or the tanycytes, right? Or what is very attractive is that there are in CSF endosomes, and endosomes, of course, contain membrane proteins and contents which are non-membrane proteins that trap these CSF constituents. And both of these, the endosomal particles, the endosomes and exosomes, I should say, this would be exosomes, sorry. The exosomes have surface markers, they have surface proteins that allow them to latch to specific receptors. And then, what I said here, we are not saying that this is that is the way the brain communicates. Of course not. It is so that, especially between the hypothalamic nuclei, but also between other nuclei and the brain, of course, signal transmission, fast signal transmission occurs via neurotransmitters. These nuclei are often very much connected via axonal connections. But this is another type of transport which I think is very important and is worth studying. So let me just finish by acknowledging the people who have done the work. When I say we or whatever one, it was usually Regina Faubel, who was a PhD student, is now a postdoc in the lab, who was adventurous enough to get working on this problem. And Christian Wessendorf, who was a graduate student of Eberhard Bodenschatz, who is a director in the Max Planck Institute for Dynamics and Self-Organization, just on the same campus. And he is a physicist and very much helped us and continues to help us in the more nitty-gritty details of doing these tracts and doing the analysis on speeds and that kind of stuff.